Hello students, now we can see the topic meiosis. The term meiosis was given by Farmer and Moore. It is derived from a Greek word meum that means to reduce. Meiosis is also known as reductional division. Why? Because it reduces the number of chromosomes to half the number of chromosomes present in the parent cell. That is, if the parent cell is having diploid number of chromosome, the number of chromosome produced in the daughter cells as a result of meiosis will be having haploid number of chromosome. Okay, that's why meiosis is said to be as reductional division. It is of two divisions, meiosis I and meiosis II. Now, where this meiosis occurs? It occurs in the germ cells of the reproductive organ for the formation of sperm and the ovum, that is the gametes. Okay, so gametes such as sperm and egg or ovum are formed by the meiotic division. Okay. Now a question arises, why the gametes are produced by meiosis and not by mitosis? By mitosis, the gametes can be produced in haploid organisms where their body is formed of haploid cells only. Okay. But in diploid organisms where the body is formed of diploid cells, only by meiosis the gametes can be produced. We can see that with an example of human being. See, in our body is formed of diploid cells, isn't it? And the diploid number of chromosome present in our body is 46. Okay, now you assume the male gamete, that is the sperm, is produced by mitosis. Then how many number of chromosome will be there in the sperm? The sperms will be having 46 chromosome. Okay. Now the same way, the female gamete, that is the ovum, is also produced by mitosis. So it is also having 46 chromosome. Now, as a result of fertilization, see, after the formation of gametes, fertilization occurs, isn't it? So, as a result of fertilization, what will be formed? A zygote will be formed. And the zygote will be having 92 chromosome, isn't it? Now, when you add 46 and 46, that is the sperm when fuses with ovum, it produces the zygote which will be having 92 chromosome. We are diploid organisms with 46 chromosome in our body, and if a zygote is formed with 92 chromosome, can it develop into a diploid human being? No, the zygote cannot exist and it cannot develop into a human being, isn't it? That's why we say by mitosis the gametes cannot be produced. Okay. Now we can see what happens when the gametes are produced by meiosis. Again, our normal chromosome number is 46, isn't it? All our diploid cells are having 46 chromosomes. Now, meiosis is occurring in the male germ cells. As a result, the male gamete, that is the sperm, is produced. So, the sperms will be having how many chromosomes? Half the number of chromosome as that of the parent, isn't it? So, if the parent cell, that is the germ cell, is having 46 chromosome, the daughter cell, that is the male gamete cell which are produced, will be having half the number of chromosomes. That is how many chromosomes? 23 chromosomes will be there. Okay. As a result of meiosis, 4 haploid cells will be formed. I am showing one gamete or one cell. Okay. So 23 chromosome is present in this male gamete. Same way, the female gametes in the female body, also the female germ cell will be developing into the female gamete. Okay. So here also which division occurs? Meiosis occurs, isn't it? As a result of meiosis, the female gamete or the ovum is produced, and here also it is having haploid number of chromosome. Okay. And at a time only one ovum will be produced in females, but in males from one cell 4 haploid cells are formed and the 4 are normal male gametes. Okay, but in female 4 cells will be formed. In that 3 will be polar bodies which is of no use, and only one only one ovum will be produced. Okay, but the resulting cell is 4 only there also. Yes, so now you assume a female gamete and a male gamete is there. Again they undergo fusion, that is fertilization, and as a result again the zygote is formed. And the zygote, you see how many number of chromosome is there? It is 46. So our original diploid number of chromosome is what? 46. Now the zygote is also having how many chromosome? 46. So now this 46, the zygote with the 46 chromosome can undergo mitosis and can develop into a new individual. Okay, so that's why in order to restore the number of diploid number of chromosome in the zygote, meiosis has to take place when the gametes are formed. Okay, that's why during gamete formation, meiosis occurs in diploid organism. Okay. Now we can see the key features of meiosis. Meiosis involves two sequential cycles of nuclear division as well as cell division. And these two sequential cycles are meiosis I and meiosis II. Okay. So two divisions are there in meiosis, that is meiosis I and meiosis II. But it is having only a single cycle of DNA replication. Before mitosis also interphase is there, and in the interphase, S phase of interphase, DNA doubling occurs. In the same way, before meiosis also, during the interphase or the preparatory phase, the S phase, synthesis of DNA occurs, that is doubling of DNA occurs. Okay, but this doubling occurs only once, not twice. Even though meiosis I and II is there, twice the DNA multiplication or replication will not take place, only once it occurs. Okay, and after DNA replication in the S phase of interphase only, meiosis I will be initiated. Okay, so before meiosis I occurs, the DNA will be doubled, and so the chromatids will be doubled at the S phase itself. Okay, the chromosome, the whole chromosome is not doubling, only the DNA will be doubling. As a result, we can say the sister chromatids will be doubled at the S phase. Okay. Then, meiosis involves the processes like pairing of homologous chromosome as well as recombination between non-sister chromatids of the homologous chromosome. Now, what is this homologous chromosome? See, in the previous slide I told you, the zygote is formed by the fusion of male and female gamete, isn't it? Male gamete is produced from the father, female gamete from the mother. When these two fuse together, the zygote is formed. The zygote will develop into the embryo and into a young one, isn't it? So each and every person is receiving one set of chromosome from the father, another set of chromosome from the mother. Okay, so from the father 1 to chromosome number 1 to 23 is received. Same way from the mother also chromosome number 1 to 23 is received in one copy. Isn't it? Not a double copies but only one copy. Now pairing of homologous chromosome means during this meiosis what happens? The first chromosome which is received from the father and first chromosome received from the mother will be pairing, will form a pair. Okay, that is these two chromosomes now is said to be as a homologous chromosome. Okay, the pair, first pair is a homologous chromosome, second pair is second pair of homologous chromosome. Each pair forms the homologous chromosome, and recombination is the crossing over between non-sister chromatids of homologous chromosome. Okay, the sister chromatid means what? The we can say the duplicated DNA becomes the chromatid, isn't it? So in a chromosome, how many chromatid will be there? Two chromatids will be there. That chromatids are said to be a sister chromatid. Now the another pair of that homologous chromosome is the non-sister chromatid. Okay, I told one paternal, that is one chromosome received from the father and another received from the mother, will be acting as a pair of homologous chromosome, isn't it? So this you assume this is the first chromosome which is received from the father, and this is the first pair of chromosome received from the mother. Okay, yes. Now this is one chromatid, this is another chromatid. These two are the sister chromatids. Okay, and this is one chromatid, this is another chromatid. These two are the sister chromatids. Okay, now this chromatid and this chromatid, we can say it as non-sister chromatid. Okay, so this we have received from the father, this we have received from the mother. Okay, so if the chromatid of this chromosome is considered, it is sister chromatid, and the chromatid of this chromosome and this chromosome, when we consider, this is non-sister chromatid, but they belong to the first pair only. Okay, now what will happen? Crossing over takes place between these two chromosomes, okay, these two chromatids, and that is what we say it as recombination between non-sister chromatid of homologous chromosomes. Okay, again we can it will be clear to you when we see the crossing over process. And at the last or at the end of meiosis II, we can get 4 haploid cells. Okay, 4 haploid cells are formed at the end of meiosis II. Okay, so these are the key points of meiosis. First is meiosis I. Meiosis I is divided into the following phases like prophase I, metaphase I, anaphase I, and telophase I. Prophase I is the longest phase of meiosis I, and it is also more complex when compared to that of prophase of mitosis. And this prophase I is subdivided into 5 phases based on their chromosomal behavior. So what are the 5 phases? Leptotin, zygotin, pachytin, diplotin, and diakinesis. Leptotin is the first stage under prophase I. During leptotin, the chromosome become gradually visible. In the picture you can see it is gradually visible, and it can be seen under the light microscope. The compaction of chromosome continues. Actually, in prophase, the chromatin will be getting contents to chromosome, isn't it? So here also the compaction of a chromosome continues. Compaction here means the condensation of chromatin into chromosome continues. Okay, and the volume of the nucleus increases, and formation of aster also begins, just like that of mitosis. Okay. Next is zygotin stage. The main important event that takes place during zygotin is pairing of chromosomes. Pairing, as I already told you, one paternal and one maternal chromosomes will pair with each other. That is 23 maternal chromosomes are there and 23 paternal chromosomes are there, isn't it? So each one will pair with each other. As a result, 23 pairs of chromosomes will be formed, isn't it? So all these chromosomes before prophase, what will happen? It will be in the form of chromatin material, and it will be seen as entangled mass of chromatin material, isn't it? Now that masses get slowly separated, and each pair will come together. Okay, earlier it is not seen in pairs, they will be seen as an entangled mass of thread. Now they come together to form pair with each other. Okay, that is the difference here. And this process of association or pairing is called synapsis, and such paired chromosomes are called homologous chromosome. So inside the cell, 23 pairs of homologous chromosomes will be formed. Okay, and synapsis is actually accompanied by the formation of a complex structure called the synaptonemal complex. Okay, and as a result of this, the synapsed homologous chromosome is called a bivalent or a tetrad. Okay, after the pair is formed, we can say it as a bivalent because a pair of chromosomes are there, and each chromosome will be having two sister chromatids. So four chromatids are there. So we can also say it as tetrad. Okay, so now that pair of synapsed homologous chromosome is called a bivalent or a tetrad. Is it clear? Here in this picture you can see the pairing. So blue colored chromosomes you assume received from the father, and the red colored chromosomes are received from the mother. Okay, just you assume like that, and you can see the first pair of chromosome here. This is the first pair of chromosome. Okay, this one is the first pair. This is one pair. This is another pair of chromosome, and this is another pair of chromosome. Okay, according to the size, okay, they will be pairing. First chromosome will pair with first only. It will not pair with the third or the fifth like that. Okay, so like this they will be pairing. So when they pair, it becomes the bivalent. Okay, because two chromosomes are paired with each other, it is called bivalent, or we can say it as tetrad because each chromosome is having how many chromatids. Here you can see two are there, and here you can see two are there. So four chromatids are there. That's why we can say it as tetrad. Okay. Next is the Paketine stage. So during this stage, the four chromatids of each bivalent chromosome becomes very clear and appear as tetrad. So in the picture you can see here, this is the homologous chromosome. Okay, one pair of chromosome. This is the homologous chromosome. Now these two together we can say it as the bivalent chromosome. Okay, or the homologous chromosome, and here their chromatids are clear and they appear as tetrads. Okay, so these are the two sister chromatids of that chromosome. These are the two chromatids, sister chromatids of this chromosome, and if you take one chromatid from this chromosome and another from this, then we can say it as non-sister chromatid. So this is sister, and this is a non-sister chromatid. Okay, so the chromatids of two chromosomes of the homologous pair can be considered as non-sister chromatids. So crossing over occurs between the non-sister chromatids of the homologous chromosome. Okay, so during this stage, recombination nodules will be formed at the site of crossing over. So this is the region where these two chromatids are crossing with each other, isn't it? So there a recombination nodule will be formed. And what is this crossing over? It is the exchange of genetic material between two homologous chromosomes. So this is the two homologous chromosome, and now the genetic material is exchanged between them because the chromosomal arms are crossing with each other. Okay, so the genetic material will be exchanged between them. This process is called what? Crossing over. And this crossing over is an enzyme mediated process, and the enzyme which is involved is called recombinase enzyme. Okay, it helps in recombination. That's why it is called recombinase enzyme. Okay, and as a result of crossing over, recombination of genetic material occur between these two homologous chromosomes. Okay, here in this last picture if you see, this is a recombinant. Okay, here you can see in the place of blue color, there it is red in color. Why it is red in color? Here crossing over has take place, and as a result of crossing over, after crossing over it will break and attach in such a way that this blue color part will be attaching with the red chromosome, and red part will be attaching with the blue chromosome. That is, a part of paternal chromosome will attach with the maternal chromosome. Paternal means received from father, will be attaching with the maternal chromosome means received from the mother. Same way, one part from the maternal chromosome will be attaching with the paternal chromosome. So that we can say it as recombination. Okay. So recombination will be completed by the end of packaging stage. Okay, and the chromosomes will be linked together at the site of crossing over. They will not be separated like this. They will be linked together, okay, at the place of crossing over. Next is diplotin stage. During diplotin, the dissolution of the synaptonemal complex occurs. Synaptonemal complex is formed at the zygote stage when the pairing of homologous chromosome occurred, isn't it? So that will be dissolving, and now the homologous pairs, that is the bivalent chromosome, tend to separate from each other except at the site of crossover, that is in the place where crossover has occurred. Only that region will be seen attached with each other, and it forms X-shaped structure there, which is called the chiasmata. Okay, and all the other region will be separating from each other. Okay, and in oocytes of some vertebrates, this diplotin stage can last for several months or even for years. Okay. Diakinesis is the last stage in prophase I. It is marked by the terminalization of chiasmata. So chiasmata is the X-shaped structure formed as a result of crossing over of non-sister chromatids, isn't it? So that will come to an end. Okay, towards the end, the chiasmata will be forming. That is, terminalization of chiasmata occurs. Then the chromosomes will be fully condensed, and the meiotic spindles will be formed, and they get assembled to prepare for the separation of the homologous chromosomes. Okay, then the other changes like nuclear membrane disappears, nucleolus disappear. Okay, and various other organelles also get disappear. Okay, these are the changes that takes place during diakinesis. Prophase I is followed by metaphase I. And during metaphase I, the bivalent chromosomes align on the equatorial plate. So in the center of the, here this is the center of the cell. So all these chromosomes, you can see, they get aligned in the center of the cells. And the spindle fibers will be attaching to the kinetochore of the homologous chromosomes in the opposite pole. Okay, see, in mitosis we have seen that if this is the chromatid of this homologous chromosome, it will be splitting at the centromere here, isn't it? So for that, if this spindle fiber is here, it attaches to the kinetochore here, and from this the spindle fiber will be attaching here, isn't it? But if you see here, it is not like that. This spindle fiber will be attaching to the kinetochore of this chromosome of this homologous pair, and from here it attaches to the kinetochore of this homologous chromosome. Okay, and the other opposite side, the other chromatid is not attached by the spindle fiber. Okay, this is a difference which you can see here. Okay, now so these changes occurs during the metaphase I. Next is anaphase I. During anaphase I, the homologous chromosomes will be separating, and the sister chromatids will be attached with the centromere only. Okay, due to the contraction of the spindle fiber, the homologous pairs will be separating. The homologous chromosomes will be separating. Okay, not the chromatids. See, that is the main difference between anaphase of mitosis and anaphase I of meiosis. In anaphase of mitosis, we have seen that. So again, I will show the diagram. We have seen that if this is the chromatid, okay, chromatid of this chromosome, this is the centromere. The centromere will be splitting into two longitudinally like this, and the spindle fiber, this is the centriole, and this is the spindle fiber attaching to the kinetochore, and this is another centriole. From here the spindle fiber is attaching to the kinetochore this side. Okay, now what happens? This spindle fiber contracts. As a result, it will be splitting like this longitudinally, and this chromatid moves to this end and this chromatid moves to this side, isn't it? So this side also one chromosome is coming, this side also one chromosome is coming. There is no reduction in the number of chromosome. But if you see this one, that is anaphase 1 of meiosis, okay? So this is one chromosome, okay? This is the other pair, other homologous chromosome, okay? And here this is the centriole, this is the centriole, and from this centriole the spindle fiber will be attaching to the kinetochore of this side, one side only, and this side is not attached, and here also you can see here it will attach and this side is not attached, okay? Now when the same thing you can observe here also, okay? Now when the spindle fibers contract here, what will happen? The whole chromosome of this pair will separate and go to the opposite pole, okay? It is not splitting here, no splitting occurs in between. Here splitting occurs, so the chromosome number can remain the same. Here no splitting is occurring, so the whole set of chromosome, that is the whole chromosome with both the chromatids will be moving to one side, and the other pair with both the chromatid will be moving to the other side. So here you can see two chromatids are still remaining with the attached with the same chromosome, okay? Like that it goes. As a result, what will be the number of chromosome at each pole? Earlier totally 1, 2, 3, 4 were there, isn't it? Now as each pair is separating, in one pole we can get 2 chromosome, in the other pole we can get 2 chromosomes. So instead of 4 chromosome, now the chromosome is 2. That means it is reduced to half. Okay, so this is the main change which take place during anaphase 1. It can be asked what is the difference between anaphase of mitosis and anaphase 1 of meiosis 1. So you should be able to explain this. Okay. So next is the telophase 1. Here the nuclear membrane and the nucleolus reappear. And after the formation of nuclear membrane, two nuclei will be formed and it will be followed by the cytokinesis. The cytoplasm will also divide and as a result you can see here a dyad of cells will be formed, that is two cells will be formed. Okay, and after the two cells are formed, so here inside the cell you can see only half the number of chromosome is there. In the initial cell there were 4 chromosomes, but now you can see in each cell only 2 chromosomes are there. Okay, and after the dyad is formed, the cells will be entering into a stage called interkinesis, and actually it is a short-lived phase. Okay, interkinesis is a short-lived phase which is seen between two meiotic division, that is meiotic 1 and meiotic 2 division. Okay, and during this interkinesis, don't think the DNA replication will take place. Already before meiosis 1, DNA replication has occurred, isn't it? And that DNA is still there in these chromosomes. They have not yet separated. So no more DNA replication will be taking place. And this interkinesis, which is short-lived, will be immediately followed by the prophase II. Okay. Meiosis II is initiated immediately after cytokinesis occurs. That is after meiosis 1. At the end of meiosis 1, cytokinesis occurs and immediately after that meiosis 2 will be beginning. Okay, in between only a very short-lived interkinesis will be there. Okay, and this meiosis 2 is just similar to that of normal mitotic division. Okay, here the nuclear membrane disappears and the chromatin material get condensed to become chromosomes. Okay, next is metaphase 2. So here the chromosomes align at the equator and the microtubules from the opposite poles of the spindle fibers get attached to the kinetochores of the sister chromatids. So here you can see in the sister chromatids. Okay, so this is the sister chromatids. So here the spindle fiber is getting attached on either side. You can see it is getting attached, okay, to this kinetochore of the sister chromatids. Okay. In anaphase 2, the centromere will be splitting into two longitudinally. As a result, the chromatids will be separated. Okay, so just similar to that of mitosis if you take. So this is the chromosome. This is the chromatid of the chromosome. Okay, this is the centromere. Yes, and the centromere will be splitting now like this longitudinally. So this chromatid gets separated and this chromatid will also be separated and it will be moving to the opposite pole. Okay, so here in this also you can see here after splitting it is moving to the opposite pole due to the contraction of the spindle fibers. Okay, the spindle fibers will be contracting and the chromatids will be moving to the opposite pole. Okay. The last stage is the telophase 2. So meiosis will be ending with the telophase 2. Here we can say 4 nuclei will be formed. So at the end of meiosis, 4 haploid cells will be formed, isn't it? So when telophase 2 is over, 4 haploid nuclei will be formed and followed by that cytokinesis occurs and 4 daughter cells will be formed and all the 4 daughter cells will be haploid in nature. What is haploid cell? Because they will be having half the number of chromosome as that of their parent cell. Okay, so we can say the 4 haploid daughter cells are formed. Okay, thus as a result of meiosis from one cell, from one diploid cell, 4 haploid daughter cells will be formed. Next is the significance of meiosis. So what is the significance of meiosis? Meiosis, we have seen that the diploid cell will be undergoing meiosis and as a result 4 haploid cells will be formed, isn't it? So what is the significance of meiosis? It helps to maintain the chromosome number generation after generation in case of sexually reproducing organism. Okay, so in sexually reproducing organism, the same number of chromosomes can be retained during fertilization. So for that, haploid gametes have to be formed by meiosis. Okay, so it helps in the maintenance of chromosome number generation after generation in the case of sexually reproducing organism. Then another significance is it can increase the genetic variability in the organism from one generation to the next generation. Okay, every time some variation occurs. Why? Due to crossing over and recombination, some variation occurs in each individual from generation after generation. Okay, and these variations are very important for the process of evolution. So that is about the significance of meiosis. Okay, thank you children.